Dr Mohammed Eslam, Professor Jacob George, Professor David Booth and Associate Professor Golo Ahlenstiel.
Well, fibrosis is a natural healing process. So it's where you get damage to ordinary tissue. You repair that damage with this healing process, but unfortunately it doesn't replace the cells, for example liver cells with liver cells, it replaces them with non-functional cells. What we found was that a gene which we had originally discovered predicted clearance of hepatitis C virus. Okay, so that gene, it turns out, also predicts, and that genetic variant also predicts how much fibrosis you have.
And we developed, using advanced machine learning technique, a test we called it FibroGene, which can predict the fibrosis with high predictive value. And this work was published early this year, and fortunately it has been included in the Australian guideline recommendation for treatment of Hep C.
Dr Chi Kao and Professor David Harris.
So currently, treatment for the CKD is limited in the number and efficiency. So our project is try to find out the new target to treatment the CKD. Our studies, we use one of the model of the CKD models. We found these cells play a pathogenic role through activated CD8 T cells. And we found if you deplete these cells, that we can attenuate renal inflammation and injuries. Firstly, they can be new marks for the diagnosis for the human CKD patients. And then this study raised the potential to target these cells in the CKD patients as a new treatment.
Associate Professor Julianne Georgievich and Dr Sophie Lev.
Invasive fungal infections are a major cause of morbidity and mortality worldwide. These infections are expensive to treat. The drugs are toxic. We found a kinase in a fungal pathogen that we work on called Cryptococcus neoformans. And this kinase we found is really important for allowing the fungus to derive nutrition from its primary site of infection, which is the lungs. So by taking away this kinase or inhibiting it in some way, we essentially reduce the availability of nutrients for the organism. So what this treatment would do, if it targeted this kinase, would be to starve it to death.